Hello, I'm going to test out this mic so I can use my hands. Hello, everyone. I'm Lena Nilsson. I'm the VP of Product here at Recursion. I've spent my entire career at the intersection of data science and human health. I have a background in computational biophysics and bioengineering. And I'm delighted to be here together with my colleague, Imran Haq, VP of Data Science, to share with all of you the story of the Recursion OS. What is the OS? It is the cumulative set of tools and processes that we have created in order to be able to build and interrogate massive data sets in biology and chemistry. It is the cumulative work of hundreds of recursionauts over nearly the last decade. Or to harken back to the introductions by Chris, Tina, and Martin, it is a scientific and technical platform that we have built, fit for purpose, to be able to industrialize drug discovery, decode biology, and ultimately get more drugs to patients faster. Today, Imran and I are going to share the details about a bunch of really cool tools. But before we do that, I'm going to take a step back and tell you about the mindset behind the Recursion OS. And this is probably the most important slide in the whole presentation, because the mindset, that really is what underpins our strategy. It's what we believe makes us unique from all the other biotechs. It's the roadmap for how we will stay the leader in tech-enabled drug discovery today, over the next year, and into the coming decades. So let's dig in. In traditional approaches, literature tends to drive discovery. You start with a narrow, focused idea, maybe a target-specific hypothesis. If you're wrong, you've lost before you even started. In contrast with the Recursion OS, platforms drive discovery in an unbiased and target-agnostic way. The way we can do this is that we look at large swaths of biology all at once. As an example, using our phenomics platform, we have charted 3 trillion, so trillion with a T, biological and chemical relationships that our scientists can then mine for actionable insights. This unlocks transformative value for us as a company, right? Because it unshackles us from the literature, from all the things that the industry thinks are right, but maybe aren't so, right? The literature has lots of insights, but it's also riddled with mistakes, omissions, errors, groupthink. And we can move away from that by starting with our platforms. And not just platforms, maybe the key word is really data. In traditional approaches, data is often treated like an exhaust. It's the output of an hypothesis. In contrast with the Recursion OS, we turn this completely around. We use data as a fuel. In practice, what this means tactically is that our own in-house data is used for every hypothesis to drive every program forward. It is data as rocket fuel to ignite and then accelerate all of our programs. But not just any data, connected data. In traditional approaches, data is often siloed within individual programs and in time. They're single use. Imagine for a second 2 scientists working on a disease. They work in different laboratories. Maybe those laboratories are set at different temperatures. Maybe those scientists run the same experiment, but in slightly different ways. Maybe they get their reagents from different vendors. If you're just running that experiment twice, you can probably get away with that. But across many experiments, across large data sets, you lose the ability to compare the things you learned, and that really limits the value of that data. In contrast, at Recursion, our data is connected across our programs and across time because of the extreme focus of standardization that we have. I'll give you an example of the value unlocked by this. Let's think about the time dimension. If I look over here to my left, your right, you see the robots in the laboratory here churning out biological experiments and data right now, this minute. That data can be compared in a quantitative way with data we generated a year ago, even with data we generated 5 years ago, the year I joined the company. And that is incredibly transformatively powerful. And I'll give 3 examples of why. One is it increases our confidence, right? We don't just see that insight, we can look at the context of all the biology and chemistry around that insight. The second thing we get is we can discover the unexpected, right? We can look at data points that maybe we would never have included if we just limited ourselves to a starting hypothesis. And third, kind of zooming out a bit and thinking, connected data is an incredible competitive moat for us. And it's a moat that grows faster and faster over time. And the reason for this is the bigger your core data set is, the more value you get out of every single data point that you add, because the more connections that data set will provide into that core data set. It's a flywheel effect. And that's something that's really hard to catch up with for others. And to build to that idea of flywheel effects, I want to add a point here around traditional approaches and learning. There's, of course, learning in all drug discovery programs, but it's often siloed within programs, and maybe more importantly, maybe more nuanced, it's siloed between the physical experimentation as separate from the digital world, like Martin and Chris both talked about a little bit. In contrast, at Recursion, with the Recursion OS, we can create virtuous cycles of atoms, of physical experiments, and of bits of our machine learning algorithms. Fundamentally, the reason for this is because we're digitally native, right? We literally built the systems to be able to do this. I'll give an example into something I'm really excited about, the progress we're aiming for in 2023. There's a lot of new machine learning approaches that really require this interplay, like Martin mentioned, between the physical and the digital worlds. One example is new progress in active learning, a type of machine learning where iterative improvements in physical experimentation are done with improvements and iteration in your machine learning models. Physical experiment model, back and forth, iterating. For a company like Recursion, the value that can be unlocked if you do this well is that you get better at knowing what experiments to even run in the first place. So you're only executing the experiments that are the most likely to unlock actionable value. That's a really efficient and powerful way to leverage your research infrastructure and your resources. And if we zoom out a level here, really what this gives us overall is a way to really rapidly learn what works and then what doesn't, and to deploy changes not just locally, but across our whole research infrastructure at once, improving the company in big leaps all at once. And finally for this slide, and this is a summary of the slide, which makes it a summary of the whole presentation. If you compare a traditional approach, the focus there is on bespoke assays and processes. You work within individual programs, focusing on a single assay at a time that reveals a specific type of insight. That's really laborious, it's expensive, it's time-consuming. And in contrast, at Recursion, what we're doing is focusing on a more limited, high-power set of industrialized assay platforms. Things that can be automated and standardized so that we can run them day after day, year after year, to continuously generate insights across a whole portfolio. What this gets us in the end, the value unlocked, is that at Recursion, we don't have to approach our portfolio of drug discovery programs as a whole series of independent parallel efforts. Instead, we have a cumulative portfolio that we can push forward with one unified research infrastructure that's efficient, it's drug discovery, industrialized. I'm going to briefly show what this looks like superimposed to a traditional drug discovery pipeline, and Chris touched upon this briefly as well. All the way over on my right, your left, you have the initiation of a program, compound hit identification, all the way out to the beginning of preparing an investigational new drug application for the FDA. In a traditional process, this is very linear. It tends to be separate teams working on different parts, using different types of assays, with data stored in different ways. This really limits the amount of learning that can happen and the amount of process improvement, and it's in complete contrast to what we do at Recursion. For example, as I mentioned on the last slide, we deprioritized bespoke assays in favor of industrialized assay platforms. We start with phenomics, the unbiased study of the morphology of cells. We layered on transcriptomics, that's the high-dimensional study of the transcript, the step between genetics and proteins. And finally, even all the way out to in vivomics, the industrialized use of animal studies. You can see a lot of the other concepts from the last slide here as well, right? Virtuous cycles, connecting data, the idea of learning across the full portfolio. And ultimately what you get, if you do all these things well, and you do them repeatedly, like we do at Recursion, something really interesting emerges. And this is new. This has not been in the introductions or in my previous slide. This is what I'm maybe most excited about the work happening today. It's what I will work on when I get back to my computer after this presentation. It's what we call at Recursion industrialized workflows. It's the idea that we are not just automating individual assays, we're automating whole sections of the drug discovery pipeline so they can run in semi-autonomous ways. Imagine what we call an industrialized program generation workflow that can automatically generate insight packets that can be handed to scientists before they ever step up to the bench, before they ever pick up a pipette, before they put in any effort or time. And that packet can give insight about which compounds are the most promising to take into hit-to-lead activities. Another example, industrializing hit-to-lead and automating processes in the early optimization of chemical compounds. This is what bringing the recursion mindset to the drug discovery pipeline gets to, and I'm super excited to be here for it. So far, I've talked about the recursion mindset and how it's a competitive advantage. I'm going to turn to the other favorite competitive advantage at Recursion in my mind. It warms my heart to be able to share with all of you that data is also at the heart of Recursion. And we think about our data advantage across three big dimensions. One is scale, just simply the massive quantities of data that we're able to generate. A second is reliability, the quality with which we generate that data. And finally, relatability, our ability to connect that data to unlock novel, useful insights. And I'm going to give you three examples of this scale, reliability, and relatability as it applies to our data. An example from biology, an example from our robotics platform, and then finally an example from data science. First up, biology, 500 billion. That's the number of neurons we produced in 2022. That's a lot. We believe that might make us the largest producer of neurons on the planet, certainly one of the largest. If we were to commercially acquire this many neurons, we estimate it would be a financial investment of $500 million or so, assuming it's even possible to acquire this many neurons commercially this quickly. To take an example closer to me personally, I worked with human cells during my PhD, and if we had taken my approach, my protocols and throughput, it would have taken us thousands of years to get to 500 billion. So I'm glad we didn't do that. And maybe the most impressive thing to me personally about this slide is how hard it is to get to 500 billion. This is working with induced pluripotent stem cells. They have to be differentiated, expanded into neurons. This is notoriously hard to do under any circumstance. And we've had to develop the processes and the in-house quality control system to get to 500 billion cells in a way that's reliable and relatable enough that we could also build our new proprietary machine learning algorithms on top of it. And that's the real magic, I think. A second example, 2.2 million. That's the number of experiments we can run every week in this robotics laboratory right here. And the thing I want you to really key in on here is that it's 2.2 million, not a static number, that's 2.2 million flux. That's the amount that our competitive advantage grows every single week. Later on, during the tours of the building, you're going to have a chance to have a closer look in here through the windows and see the robots working. And when you do, I invite you to imagine for a moment all of that complex robotics infrastructure in an orchestrated dance with an equally complex digital infrastructure that you just can't as easily observe in order to execute those 2.2 million experiments, over 10 million associated microscopy images every single week. And what do we get from all of that? Like Chris mentioned, a massive proprietary data set of over 21 petabytes of data. I'm told that is more than all the feature-length movies ever made on the planet, in any language, in 1080p resolution, times 5. It's a lot of data. And like Martin mentioned, we all are appreciating today, right, the incredible central role of massive data sets in enabling modern machine learning. In internet applications, like in DALL-E and ChatGPT, we're all seeing the incredible outcomes that are underpinned by massive data sets. And that's exactly what we see also at Recursion, the insights we can draw from massive data sets. And maybe the big difference I would call out relative to many other applications, it's how high the bar is for biotech applications when you talk about the reliability and relatability needed. The reason for this is how complex biology is, how much of it is unknown by humankind. You can't have a control set so easily. And also, as a third point perhaps, just how important it is to get it right when you're talking about human health. And that's exactly why we at Recursion have invested so much effort over the last decade, not just to create a massive data set, but to create a data set that is reliable and relatable. So far, I've talked about data kind of generically, and I'm going to transition here to tell you how we turn our data into data layers and into maps of biology and chemistry. If you don't work at Recursion and you don't get to play with this data directly, this might be kind of hard concepts to wrap your mind around initially. Like, what's a map of biology? What do you mean a data layer? So I'm going to use an analogy to explain what this all means. And the analogy is mapping and navigating the physical world around us. Let's imagine you're standing at this intersection. You'd probably be able to tell me the number of houses there, what types of houses, the species of plants, perhaps. This is analogous to running one high-throughput assay in biology. You will get lots of reliable readouts, but they will be limited in the type, right? Let's go back to the intersection. Let's say I now asked you, where in the world are you? You probably wouldn't be able to tell me. You might be able to surmise it's somewhere tropical. I see some palm trees, but that's probably it. But let's zoom out a layer. Now, you would be able to tell me about the shape of the road grid, the topography of the terrain. And if you have been in this location before, if this is a known geography, you might even be able to tell me that this is on the outskirts of Honolulu, Hawaii. However, if this is somewhere novel, or novel biology, you might need more layers in order to be able to say with confidence where you are. If you're like me, you've never been to Hawaii, you might be able to need to see the whole state's island chain before you can say that with confidence. The take-home of this, through this analogy, is that I want to emphasize in on the power of being able to densely connect data within a data layer, tell me about all the species of plants, but also the confidence and context you can build by moving between layers. Where in the world am I? And that's exactly what we're doing at Recursion by building lots of different data layers, lots of different maps of biology and chemistry. So for example, I've talked a little bit about our data layer in phenomics, in transcriptomics, all the way up to in vivomics and the study of animals. In fact, adding data layers like this, that's a core part of our strategy. We have ambitions to add layers in proteomics, eventually also in metabolomics, potentially, like Chris mentioned. I'm going to step out of this analogy and I'm going to invite Imran up to give us a real-world example. We're going to zero in on the phenomics layer, our phenomics maps of biology and chemistry. And Imran is going to show us how that plays out with real-world data of both known and unknown biology, known and unknown chemistry, and how a drug discovery scientist might navigate through that to pull out some interesting learnings. Imran.
Thanks, Lena. I'm Imran Haq, VP of Data Science here at Recursion. By background, I'm a computer scientist by training, but my computer science friends call me a biologist. I spent about a decade in the biotech industry, in diagnostics for inherited disease, for cancer, and my biology friends call me a computer scientist. And I'm not exactly sure what to call myself, and that's why I'm here at Recursion, because I think that shared vision, that bringing together computer science and data, as well as biology and chemistry, is really key to unlocking the power of biology for human health. As Lena mentioned, what I'd like to do now is give you a more concrete view into how we apply our mapping and navigating technologies to actually discover drugs here at Recursion. You've seen some of the analogies and some of the principles that guide our data collection, collecting data at scale, collecting it reliably, and collecting it so that all those data points can be related to each other. Now let's see a worked out example. And let's do it with a disease that I think will be more familiar to the crowd, and that is breast cancer. So our maps of biology and chemistry are, broadly speaking, keyed by genes and keyed by compounds. So one of the things that a drug discovery scientist at Recursion might do is to start with a disease that's of interest to them, say breast cancer. You could then go to an external data set, say the human phenotype ontology, and find all of the genes that are associated with breast cancer. If you do that, you'll get the yellow edges that are listed in this graph up here, where you see a number of well-known breast cancer-associated genes, and then you start seeing the relationships that emerge in Recursion's map. Those are the green edges between those genes that are around the periphery. If you look closely at that list, you'll start to see a number of patterns emerge. For example, right here above breast cancer, you'll see a very tight cluster of the BRCA-associated genes. You'll probably have heard of genes like BRCA1, BRCA2, PALB2, RAD51, BARD1, all of which are strongly associated with breast cancer, and all of these show up as an extremely tight cluster in our data. Now, one thing that's critical to realize is that we didn't have to encode that into our systems. This all fell out naturally from the data that Recursion collects. We performed knockouts of each of these genes. We imaged we imaged the cells with those knockouts on our microscopes, put them through our data pipelines, and these tight associations immediately came out. You'll see other clusters show up as well. So for example, another set of well-known cancer and breast cancer-associated pathways, RAS-RAF, HER2, PI3 kinase. Again, these are discoveries that took decades and hundreds or thousands of scientists to discover out there in the world that Recursion was able to immediately recapitulate from an automated analysis of its data. And we think that that's why the scale, reliability, and relatability of our data sets is so critical to what we're doing here. But if all we could do was look at genetics, this would be interesting, but not sufficient to transform drug discovery. And one of the really unique attributes of Recursion's platform is that we don't just see relationships within biology or relationships within chemistry, but we can actually see the relationships between the two. So let's zoom into one of these clusters to understand what I mean. And we'll zoom into this RAS-RAF, HER2, and PI3 kinase group. If we zoom into just that set of genes, what we can do is put up a set of well-known compounds to validate that the relationships that we're seeing in our platform are those that you would expect. So for example, at the bottom, you'll see a couple of well-known RAF inhibitor drugs, dabrafenib and carafenib, and you'll see that they have the relationships that you would expect to genes like BRAF. You'll also see other investigational stage molecules, AZD4547 and others at the top, and you'll see that they're making the relationships that you'd expect. These were originally developed as TGF-beta inhibitors, and you'll see that they have relationships, again, falling out automatically from our mapping and navigating data with genes in that pathway, like TGF-beta receptor 2, right there, right above breast cancer, FGFR1, and so on. So what the relationships on the last gene or on the last slide, and the relationships on this slide should do, is give you confidence that what our approach is doing is recovering real biology. We're able to recapitulate not only the history of genetic discovery in breast cancer, we're also able to recapitulate the history of drug discovery in breast cancer, or at least in this pathway in breast cancer. But if all we could do was recapitulate history, that wouldn't necessarily show the way forward. So you'll notice there are a lot of gaps in this diagram. Let me fill them in for you. What we have now is the rest of the compounds in the recursion data universe that show strong associations with the set of genes in this pathway, with these RAS-RAF genes. Now, I haven't labeled them because these aren't approved drugs, these aren't clinical stage drugs. These are starting points in Recursion's chemical library that we have screened that are also showing similar associations to these genes. So imagine for a moment that we lived in a world in which we hadn't done 25 years of research on the TGF-beta pathway, that we hadn't worked on EGFR and HER2. If we were in that world, we could take any of these starting points and then initiate research programs against them in order to drive towards drug discovery. And this is the real power of the Recursion platform. Not only do we recapitulate massive amounts of biology that we already know about, we discover new biology that was not known about or that was erroneously marked as not existing. We can then put that in relationship to large numbers of chemical starting points in order to initiate and advance drug discovery programs in the way that Chris and Lena have mentioned to you already. But this, of course, is just a small view of what exists in the Recursion data universe and in Recursion's maps of biology. This is zoomed into about a dozen genes. So let's zoom out. Now, as Lena mentioned, we have over 3 trillion relationships in our maps of biology. This projector has maybe a few million pixels on it, so we're about a factor of 1 million away from even being able to represent all of these relationships. So this is a very schematic view of what you might see if you were able to zoom out into Recursion's maps. You see associations between diseases and genes in yellow. You see associations between genes, showing you how biology is put together in green, and you see relationships among genes and compounds in orange and purple, showing how all these pieces are put together in order to enable Recursion's scientists and Recursion's algorithms to navigate the space of biology and chemistry in order to move forward towards drug discovery programs. So in order to make that translation from maps to programs more concrete and show off some more of the technology that we've built to progress not just from relationships in the map, but really progress to that IND enabling phase, I'd like to invite Lena back on to go a layer deeper in the Recursion OS.
Thanks, Imran. So Imran just gave us a specific real-world example from within a map of biology in phenomics. You actually have 2 slides left. Do you want to come back up, Imran? He tells these 2 way better than I do.
That was a teaser. Now you get to learn a little bit more about the Recursion maps. So one point that I'd like to emphasize is what Lena mentioned about the way that data is treated as an exhaust in a traditional pharma setup versus the way that Recursion approaches data and approaches its direction to doing discovery. In a traditional target-based screen at a pharma, what you might do is look in the biological literature and identify what's a target that you would actually like to that you would like to go after in order to treat a particular disease. You'll then establish a unique experimental setup, a unique assay or assay platform to screen that particular target, and then perform a high-throughput screen against that one target in order to identify the chemical matter that you might then progress to hit to lead optimization, and so on. Now, once you've done that, you've gotten a huge amount of information. You know, against that one target, what do all of those 1 million compounds look like? The problem is that's not nearly enough information in order to efficiently progress drug discovery downstream for a couple of reasons. From a scientific perspective, when you get that information about that one target, you're missing out on a vast amount of information against other targets. Polypharmacology is a well-known phenomenon in drug discovery and therapeutics today, where drugs don't have effects on just one target or one protein in the body. And in fact, the effects on multiple proteins may not just be important for safety in the sense that off-target effects might lead to toxicity, but can actually be critical for efficacy as well, where modulation of multiple targets is actually necessary in order to drive efficacy against a particular disease or to drive differentiation of where different subtypes of disease may be affected by different drugs. Beyond the scientific reasons, there's also a critical financial or efficacy or efficiency standpoint here as well. If you're interested in going after one target, great, you've got information about that one target. But when you want to stand up a new drug discovery program, you're now having to find that target, establish an entirely new assay, run the same screen again, and so the costs start to build up as you start to build that pipeline. That's not the kind of thing that you can do if you want to have that ultra-broad pipeline approach that Chris mentioned at the beginning. Recursion's approach is different. Because of the technology that we've built in our mapping and navigating approach, when we run a screen on our phenomics platform, each of those 1 million compounds that we screen can automatically get put into relationship with all of the gene knockouts that we have run, all of the compounds that we have run, all of the soluble factors that we've run. So what that means is that every time we run a screen, every compound automatically gets put in relationship to that entire library of gene knockouts, and we can model its activity against not just the one target, but actually all of the potential targets that we might be interested in. And that's the fundamental change that's able to turn drug discovery into a search problem, and in particular to turn it into an unbiased search problem, where not only do we find out if a compound was active against one target that a scientist may have been interested in, we can automatically find out whether or not it may have toxic effects, if it's showing up against other proteins that we don't want it to inhibit, or potentially if it affects multiple targets that may be important for the modulation of disease. So at this point, I think these are the two slides, and I will actually hand it back over to Lena.
So at this point, Imran and I are going to zoom back out and show how these different data layers, including the phenomics maps of biology and chemistry, are layered onto a drug discovery pipeline. I'm going to talk to you about the phenomics layer, give an example from transcriptomics, showcase a bit of our in vivomics work, and then I'm going to hand it back to Imran to show how we layer on chemistry on top of all of this work. So first up, phenomics. I'm going to give you a recipe for how we use our phenomic maps of biology to initiate drug discovery programs. First off, you have to actually physically have the experimental map. I would argue that the hardest part here is to model disease. Imran gave us an example using CRISPR-Cas9 to knock down genes across the entire genome. We also have lots of other perturbation technologies at Recursion that we build, we're currently working on, both in genetics and in other areas, like soluble factors or the use of toxins. Once you have your physical map, you need to capture a signal from that map. That's what happens here in the robotics platform, including in our microscopy stations, automated microscopy. And you're going to have a chance to dig into that later as part of the tours of the building. Next, you need to be able to detect and differentiate between cellular states, between healthy and deceased, using machine learning algorithms. Lots of companies say they do AI, and it can be hard to know if that's real or not. And I'm really excited to be able to share that at Recursion. You can go to rx.ai. You can download millions of images from this robotics laboratory, as Chris mentioned. You can download features that have been creating using our machine learning algorithms. You can look up papers and preprints and really dig into the way we work, dense nets, gradient reversal, the way we do batch normalization, and so on. So I invite you to do that and learn more about how we do machine learning at Recursion. But then finally, of course, having machine learning features, that's not the end goal. The end goal is to have insights that are useful for our drug discovery programs. And we do that in two main powerful and complementary ways. One, you heard Imran talk about. We create tools, web apps, that our scientists can use to mine that data. And later on today, you're going to have this chance to see a demo of another piece of software we use to also explore this data in order to extract actionable insights. The second way, I mentioned early in this talk, that's these industrialized workflows. That's the ability, for example, to find, verify, and validate insights from the map automatically without massive scientist intervention and effort. And one way we do this is to layer on transcriptomics, and I'm going to use this as my second example. Adding on transcriptomics in order to select which compounds we should put our effort in out of all those millions from the maps in our hit-to-lead activity, into chemical optimization, into ever more efficacious and safe compounds. The basic way this works is first we have a phenomic map of biology and chemistry. We run secondary phenomics experiments to verify the results. We then layer on transcriptomics, so an independent biological measurement, in order to ultimately get more confidence and more understanding about what compounds are the best to pursue and why. The transcriptomic layer here, traditionally, transcriptomics is often run sample by sample by sample. It's very expensive, it's time consuming. And what we've done at Recursion is build a robotic version of transcriptomics using multi-well plates where we can look at lots of compounds at the same time. It's conceptually analogous to the phenomics laboratory right here. In fact, we just rolled out a dedicated transcriptomic laboratory space just across the road behind me here in the building next to us. And what we can do there is run some 15,000 exomes every week. And we actually have technical capacity to run much more should we decide to turn on the tap for more of those. Why is this valuable in practice? Why does the 15,000 matter? It means that we can create dense connected data layers also at the transcriptomic level. If we see something interesting in the maps of phenomics, we don't have to pick our one favorite compound or even our dozen favorite compounds. We can look at hundreds of compounds at the same time across many concentrations with lots of controls and compare it with lots of other biology of relevance. This means that we get both a nuanced and at the same time very broad net to help us decide what chemical matter is really the most likely to be successful at later stages. And with that, I'm going to transition to my third example of a biological data layer, in vivomics. In vivomics is the use of video in high temporal resolution animal temperature in order to run high-dimensional animal studies. And at the very surface, switching here to talk about animals and in vivo, that might sound like a big transition and break from the rest of this presentation, which has been based on the in vitro. But it's really the exact same story, right? Phenomics, that's the unbiased, high-dimensional readout of the observable characteristics of cells. In vivomics, that's the unbiased, high-dimensional readout of the observable characteristics of animals. It's also the recursion mindset showing up all over again. It's an industrialized platform. It's virtuous cycles. It's connected data. We're just now layering in the in vivo efficacy, tolerability, and liability insights. So let's quickly in vivomics to traditional approaches. Over here on my right is just one bespoke assay I could show you. It's a rotor-rod study. These have been run in the 1950s. They're a popular way to measure the mobility of mice by putting them on these wheels and seeing how they do. In contrast, what we do at Recursion is run in vivomics. Over on the left, it's 24/7 capture of video of the behavior of mice in their original cages without handling and effects by human handlers, in order to extract out in an unbiased way the signals that really differentiate, for example, the behavior of a healthy mouse and a diseased mouse in a way that is quite nuanced and doesn't require us to make assumptions about what signal is important and what isn't. It gives us a lot of value. One thing that's different is that we have continuous data generation. We don't have to wait for weeks to the end of a study to know if it worked or not. We can begin already on day one to begin to predict what's going to happen and what we should prepare for coming up. Many of the other benefits you see here are the exact same ones we saw for our other data layers. It's high-dimensional assays, they're unbiased in their readouts, we save time, we save money. All great stuff, but abstract, right? So let me give you some example what this actually means in our vivarium. So we have transitioned our entire vivarium, our animal facility, to be a digital vivarium. Over 1,000 cages equipped with in vivomics. And we're going to give you 3 examples of the kind of studies that result. First up, in vivomic-enabled disease induction. Disease induction, that is ensuring that your animals really have the disease model you think they have, and then if they do, that you're able to detect that. That might sound like a trivial problem at first, but it's really not. Many diseases show up with much less than 100% population penetrance. That means not all the animals will get the disease. And second, even if they do, that disease signal can be really subtle. Lots of diseases cannot be readily studied using animal models. You don't quantifiably and repeatedly pick up on the signal. And this is exactly the problem that our in vivomic disease induction solves. What value does this give us? One, it lets us reduce the time, but quite a bit, in being able to set up new efficacy studies. And not on this slide, but what I'm really excited about, it also allows us to study diseases that have been previously thought not possible, not tractable using animal models. And now this whole disease space is open up for Recursion to pursue. My second example is digital tolerability. This is a method for figuring out which compound and at which concentration you should run in your efficacy study. Efficacy studies can be quite time-consuming and they can be expensive, so you want to be sure that you have exactly the right substrate that you're testing to get a high confidence on if this is a good path to go down or not. And that's exactly what our digital tolerability studies allow us to do. And importantly, they do that without delaying the onset of us being able to kick off that study. It doesn't slow us down at all. And then my final and last example here, liability in vivomics. When you run a compound in an animal, you test a compound in an animal, you obviously care to know that it works, right? That it's efficacious. But you also want to know that it's safe. And that's what liability in vivomics gets us. It allows us to compare the signature, the high-dimensional in vivomic readout of our compound that we're testing with known liabilities. For example, different types of liver toxicity or different types of kidney toxicity and so on. And that is also really powerful and something that we can do in a very efficient way. I've just now told you about 3 different biology layers, phenomics, transcriptomics, and in vivomics, and I'm gonna hand it over to Imran to tell you about how we layer on chemistry on top of all of these processes.
So at this point, you've heard about the deep biology platform that Recursion has built, ranging from in vitro cellular manipulation and screening, with morphology, with transcriptomics, and in vivo data. The platform that we've built is incredibly flexible. We have chosen to do small molecule work on this as our primary aim for drug discovery, but we've also targeted this platform at a range of large molecules and biologics. We've done siRNA work, we've worked with proteins, we've worked with monoclonal antibodies. But chemistry is different, and the way that I'd like to communicate how different chemistry is, I shared what computer science friends and biology friends think of me. Despite literally having done a PhD in computational chemistry and cheminformatics, not once have the chemists called me a chemist. And in order to show the ways that Recursion is addressing those high standards that chemists and pharmacologists hold themselves to and hold their products to as their molecules move forward into the clinic, I'd like to share some of the work that we have done and that we aim to do in advancing chemistry capabilities in the Recursion OS. Our vision is, just as we've done in biology, to integrate both digital and robotic operations in order to advance chemistry, in order to try to reduce our time to candidate selection from months to weeks. Now, you may be familiar with a notion in medicinal chemistry and drug discovery known as the design, make, test, analyze cycle. First, a chemist or an algorithm will design a molecule. That molecule has to actually be made or ordered from a catalog. You'll then test that in a suite of different assays, whether they're biological assays like those that we run on our platform here, or other assays that you might need to run for ADME tox or DMPK reasons. Analyze all of that data, and then design the next round of molecules to go through that process, hopefully increasing your understanding and the quality of the molecules at each step. Our approach is to develop technology in each one of the phases of the DMTA cycle in order to improve its efficiency in 3 different ways. First, to reduce the number of cycles that we have to make around this loop in order to reach the molecule that will be a clinical candidate or that will be a development candidate, or to terminate the program. Second, to shrink the time required for each time around this cycle. Time is critical. We would like to prosecute a broad portfolio of drug discovery, and speeding that up makes a difference. And finally, to increase the throughput at each round this cycle, to be able to test more molecules at each step in order to maximize the chances that the programs that we advance and the molecules that we advance are the right ones for the patients downstream. So let's talk about the ways that we are making advances in each of these steps, starting with the design and analyze processes in our digital chemistry platform. We've talked a lot about the things that we have put into production here at Recursion, and I'd like to highlight a little bit of the research contribution that we've made as well. In particular, a pair of papers that were presented at workshops at NeurIPS 2022 in December, arguably the premier conference in machine learning, show off the work that Recursion's research scientists have done to push the bounds of research and the bounds of advancement in chemistry and chemical machine learning. One paper on what are called multi-objective G-flow nets discuss our advances in generative chemistry, how we can have algorithms design new molecules, select new molecules to enter this DMTA process. As I mentioned earlier when talking about the map, drug discovery is intrinsically a multi-objective problem. There's not just one target that you care about, there may be multiple targets, and then you also have to think about considerations like the pharmacokinetics or the metabolism of this drug. Generative chemistry algorithms, therefore, need to be able to optimize multiple parameters. And the algorithm devised by Recursion scientists, multi-objective G-flow nets, outperform other benchmarked algorithms in numerous multi-objective settings in chemistry. On the analyze front, we've built a machine learning model that we call MOLE, a molecular foundation model for drug discovery. A challenge in drug discovery, as compared to other domains of machine learning, is that the amount of data that you have is actually quite small. We talk about 21 petabytes of data, but when you're at the beginning of a cycle for optimizing a set of compounds for a particular drug, you may only have a few dozen or a few hundred data points. That's not that many to go off of. And so what you need are machine learning models that can learn from history and then very efficiently learn to predict properties on the local series that you have for a particular problem. What our scientists have done is adapted methodology and technologies from large language models that power things like ChatGPT in order to train what we call a few-shot learner in chemistry. And this model achieves top 1 or top 2 performance on about 2/3 of the ADMET tasks in a public benchmark dataset called the Therapeutic Data Commons, including all of the distribution and metabolism tasks, and is the top performer in 9 out of those 22. We've already started to use MOLE and G-flow nets to power the internal drug discovery processes and the virtuous cycles in our programs. Now, beyond the contributions that we're making on the digital front, we're also building advances on the physical and the robotic front of chemistry. Behind you and to your right, we have our brand new automated DMPK testing facility, a Recursion proprietary design to automate the execution of 3 critical DMPK assays on hundreds of molecules per week. That's enough bandwidth to not only get the data that we need on the programs that on the molecules we're progressing for our programs, but also to provide additional data to fuel our machine learning, to turn these traditionally extremely difficult tasks in ADME and tox into predictable problems that we can solve using our models. And finally, on the make front, Recursion has ambitions to build automated microsynthesis systems, where we believe that automation of chemical synthesis will enable broader SAR cycles, broaden the number of molecules we can test at each point in the process, and enable a faster turnaround time for active learning cycles. I want to return to that concept of active learning that Lena mentioned. The notion here is that you can have a computer algorithm pick what are the next places in space, in chemical space, the molecules that you might test, in order to not only optimize the molecules, but optimize the model's understanding of the problem in order to pick the best molecules downstream. Now, as I mentioned, drug discovery is an intrinsically multi-objective problem. Active learning has been of interest in the pharma industry for literally decades. But I believe that the multi-layered data that we're building here at Recursion doesn't just empower drug discovery scientists, it empowers our algorithms. And our goal is to automate and industrialize as many steps in this process as possible. And the breadth of data that we collect is truly key to enabling that, because you can only optimize those pieces that you see. That's why we build scalable, reliable, and relatable data sets across these multiple layers of biology, across the morphology and the transcript levels of individual cells, moving up to the DMPK properties of molecules, and indeed the entire organism level properties that we assess in vivomics.